Dr. Ruben Abagyan, founder of Molsoft LLC, who will be speaking about ICM as a comprehensive environment for drug discovery, live on Teams from the US. Dr. Ruben Abagyan is a professor at the Skaggs School of Pharmacy and Pharmaceutical Sciences, founded Molsoft, became director of computational chemistry at the Novartis Institute in La Jolla, and spent 10 years as a professor at the Scripps Research Institute and 15 years as professor of pharmaceutical sciences at UCSD. Let us hear Dr. Ruben, everyone.
Good morning, everyone. It's a great pleasure, dear colleagues. I will make a relatively short, introductory talk to your meeting, and I will basically talk about transformations of in silico drug discovery pipeline, which we've done with Molsoft, as well as my some of my academic research as examples of how this software can be used. And so I think, first of all, I want to thank Sarojit and his great Advent Informatics team, and a number of the key companies which were using ICM or already wanted to use ICM. And so I think without you and without Andy traveling to Bangalore, I think so that the meeting would not be as great. But I think it's a remarkable, and ultimately our goal is to interact, and through that interaction and through collaborations, through working together and through listening to each other and taking seriously each other, I think that we'll make substantial progress. I will start from there is no, Molsoft. I think Preet already said a few words on Surajit as well, so that Molsoft is a special company. It started in 1994, very early on. I don't have this timeline. I think Andy will show you maybe that slide. We started in 1994 when we moved from Germany, and the rights to the software were given to me by the European Molecular Biology Lab. That's the pan-European kind of organization main in molecular biology, because their policy was to give the rights, copyright back to the authors. And that's when I moved to New York. I started a company, and I think that's how it began. And now the key element of the key invention and mathematics, and of course I will not bore you with mathematics. It's all published in multiple papers. The key advantage was that we wrote the equations of how to model a number of like any number of molecules, which are described by internal coordinates, basically as robotics. So there are connections, and there's certain length, and there's certain angles instead of the independent coordinates, because by doing that, the main goal was to enable sampling of the space, basically going through the space in such a way that we can very quickly sample all the needed conformations and sample effectively. And we developed this global optimizer, which was quite smart strategy of that search. And that algorithm became the first, the one which actually solved the flexible docking problem. So algorithms before us, they could not do that, and algorithms after us, all of them, they imitated our strategy, including the some of them. For some of them, it took 10 years. For some of them, it took 5 years. But they all basically were trying to reproduce. But certain secrets remained with us and with Molsoft, and we continued to lead in that direction. So the ICM, the internal current mechanics, and some of the algorithms which you don't really see, they happen invisibly for you in the back end of the of the screen, but that they return you they find from any random starting point, they find the right answer. So and here we're also highlighting the covalent. So these are all the kind of benchmark and important subdirections of that. So covalent benchmarking. and large screen catalog. So that's about internal coordinate mechanics. And as just as one example, of course, we work with many great companies, but in pharma, in big pharma, medium pharma, small pharma, and most importantly, with you also now as we're very proud to be working with you. And this is an example. This paper is an example of how ICM was used in Novartis for many years, and they continue to use that. And basically, it shows that the ICM Pro not only can be used as a kind of out of the box, but also can be configured to your specific environment. And in Novartis, they call this environment built with ICM and around ICM as focus. And that's the paper documenting that every day 200 chemists, for example, were doing designs. And until now, I think the majority of the drugs which came through the structure-based drug discovery, they were visualized. They were, some parts of the discovery was in the computer with ICM. And actually, on the very documentary about the imatinib, the BCR-ABL inhibitor. So the scientists in the Novartis were looking in the ICM visualization to actually look at the imatinib. So Max Stotroff and Eugene were among the first to join Molsoft, and key people there. What's important here. What I want to say is that first of all, it's a configurable software, and secondly, it's a special company because we're a small company, and for that reason, we're stable. So since '94, the company is in. It was the same people. Andy joined very early, and Andy is a great scientist. He published also many key publications with ICM. But if you look at any other company, they may come and go. They may grow to a very large size, but they cannot support themselves with their sales. But Molsoft is different. So we're stable. We're there since '94, and we continue to be there. And the main assurance of that is that we continue to innovate and continue to keep up with the time. And so this slide shows that ICM is all in one. So there are different layers, and it is also ported to different operating systems and different hardware platforms. Like, why is that? How can we, with a small team, do that? Well, we do that by consistency, by really high professionalism, and the same people doing that for many years. So we know the code in and out. That's why we can take and just completely, translate it into a different platform, or maybe take some algorithm and convert it into GPU. So that ability is important. So let's see what are the layers. The first layer is your front end ICM. It's not just visualizer. It also can compute most of the things, except for the large screen. It's just inefficient to use only your front end workstation. But you can do most of the things using the ICM Pro front end. And then the Focus is another name which was used in Novartis because they also wanted to modify some of the menus. You can do the same. You can have your custom menus in the main ICM client. And so this workstation thing is full functionality, and that workstation will work on Linux, on Apple, on Windows, on all these platforms. And it's so this is another big advantage, is that it is cross-platform because we can do that because we know the code, and it's one code wrote with the same coding standards by the same people. The back end is another layer. Back end of ICM is CPU and GPU modules, which don't need the front end. So they do some calculation with basically the same engine as runs the front end. Probably I can make a pointer right here. Let me try to do that. Pointer, laser pointer. Yes. Can you see my pointer? Yes, good. So, it has all the things which run in the background, and ICM can launch that can launch that thread, and the threads can run. And the GPUs are particularly interesting because GPUs can, in parallel, do 16,000 different things on a one little box. So this is the backend, and then there's also the uniform format, like the unified single file exchangeable ICB file for ICM. Of course, ICM can export in multiple formats, can read multiple formats, but its internal format allows you to make one document which has all the details, how you like to see it, how, so-called slides, which we call. You not only can generate some current image, and you can also automate it and run a script which will save them and put them into a movie, but you can also save the whole file with multiple objects, process in a certain way, visualize in a certain way, tables and alignments and sequences and all these elements all in one file. Then you open it and you see it exactly the same way as your colleague can see. So this ability to exchange those documents, just like you exchange PDFs or docx files, is very powerful. Yeah, so this is the all-in-one thing, front and back end and the rest, and the changeable file formats. Now a few words about research. Well, so basically we're just curious about structure-based drug discovery. I mean, the when we started, it was before even human genome was determined. It was in '94. There were tiny PDB collection, and then no genome. Now, after the genome was solved, now we what we want to do is we want to understand what is the disease. Now we understand the full flow because we know all the genes. We know how they, so we want to understand the disease, basically. We want to understand the pathology. And the pathology emerges from, okay, something is wrong with splice. Okay, let's fix the splicing. Something happens with the protein. Maybe it's mutants. Maybe it's post-translational modifications. Maybe it's homologs, expression, activation. Then each of the structures will have multiple functional states. We want to capture that. The malfunctional states may have different conformations. Maybe you want to discover a drug which only hits the inactive state, but not the active state. Transient interfaces, maybe you want to stick something in that. And then ultimately you identify the pocket, which can be between the proteins, maybe between 5 different proteins, but it's the pocket, and that's ultimately the target. And then it goes into pathways, and sometimes with a single chemical, but you can design with ICM something which hits multiple points in the pathway. And we published along those lines as well. So now, in terms of ICM, that's where we are. We identify this complicated target, and we define not just one target, we define the target profile, which means that I want to hit that mutant and this other mutant, but not the wild type. And it must be in the interface with this different protein, and that's your target profile. Then you want to screen a chemical space, and now we want to screen giant chemical spaces, which no one can do because it's too difficult and requires special acceleration. And you want to do it in a rigorous way, not in a poor docking method, but in a solid docking method. And then here, either screen the chemical space or generate in that space. If the space is so huge that you cannot even enumerate it because there are too many molecules, then we now have several methods which generate molecules in that space. Then we want to select the top large number of candidates. We want to more carefully dock them, more carefully optimize them, re-rank them with taking multiple other activities into account. And then, so then it goes into the experiment or experimental collaborators, and then ultimately it leads to the discovery. And so this is from one of our recent patents. which where ICM was the key to generate some new replacement for venetoclax, which hits the venetoclax resistant mutants. So that's our ultimate goal. Now the challenges which ICM now is pushing through, and there is no time to review all the functionalities, but these are just the new challenges, right? So first is the improving the target model, and the target can be as complex as the complex of 5 different things with in a particular state. And you need to improve the model and activity property profile. Sometimes we have everything for the model and maybe have too much information for the model, for example, multiple structures. Sometimes we don't have any. So we need to improve that to start the screening. Then, most importantly, we want the most accurate docking and scoring algorithm of the hits. That's hello. Then we want to be able to screen new chemistry, and then use the GPU acceleration. And this is the mole screen, which Surajit mentioned, which is a large collection, several thousand, quite sophisticated models, which combine multiple three-dimensional structures, different machine learning methods, and then AI as well in some of them to predict activities and other targets and properties of your top hits. So that we will use that to annotate. So ability to generate or screen is key. Now, in order to dock, I already talked about the sampling engine, but another element of that is the score. How do you score your molecules? And the scoring is ideally it is the free energy of binding which you want to predict, and the score should be should correspond to that, not necessarily in value, but in order. And so here's this RTCNN, which we published not a long time ago, which we now use in a mixture with the physical score. This is also physical, but it's trained using the neural networks. And RTCNN is NN in RTCNN stands for neural network, and you can see how much improves that score, the recognition and enrichment on a benchmark. So here's that's an important slide because it shows you where we are with accuracy of docking. And this is simple docking. This is self-docking of a benchmark. And we were reminded about that by Charlotte Dean's paper on postbusters, where she said, well, let's look into these deep learning methods, which were uh, uh, kind of discussed. And this is what AI, sometimes equated with AI, but not necessarily, but deep learning versus the classic, which is like ICM. And you can see that deep learning, uh, hello? You want to say something? No, no, okay. Yeah. Okay, good, good. So you can see in Charlotte's Dean, so this is from her paper, the best accuracy, the best fraction of correct poses by her definition and was 12% with deep learning. So-called classical methods like Vina and Gold, so that will be 58%. Molsoft jumps right away out of the pocket, out of the box, 80%. And now we're publishing a paper. We analyze this remaining 20%, and it's quite clear what's happening. In many cases, it's just a problem with the benchmark. So actually, we're much higher than that. So basically, having the most accurate docking method and scoring method is key, because otherwise, why would you start screening if you don't have that method? You will just miss most of the right solution. Of course, you'll get something. Even with 12%, you will get something, but it will not be as good and it will not be as many. You will not find that unique solution. Now, another element of this is data, and we serve many data, and one of the data sources, which is intimately connected with ICM and its models, is we call the pocketome. The pocketome is a collection of all the human and non-human proteins which are superimposed around the ligand. So the protein is the same, but the ligands are all different. And so that collection of those, you can search for ICM directly. You can get them for your project. For example, you suddenly jump into a new project. So you look up if you have this pocket of entry for your project, and you can find it. So it gives you two sources. One is a set of experimental structures, experimental for the pocket for your protein itself, and the second is the pharmacophoric features of the ligands which are co-crystallized with that, or maybe determined by cryo-EM. And so we, with that, we do all kinds of things. Most important one is that this collection becomes the entry point into our MOL screen, which are the kind of models which have it inside, but you don't necessarily see it. It uses all this information. Another enhancement to docking is ability to dock not just to the pocket, but also uses cloud as the set of features, pharmacophoric features, and then the pocket is additionally held by this distribution. And you can see in the blind prediction the accuracy which we achieved. This was published in the evaluation of this docking competition. So that's one way to enhance. Another way to enhance is, and something we're currently working on, publishing basically results out there, is that you can start. Well, I see I'm now connected with MD, so you can start from some confirmation, and instead of just running MD where it generates lots of confirmation, you can trace for each transient confirmation, you can trace the RTCNN score, and you can pick the improved RTCNN score, and you can screen with that confirmation. So that shows that with that trick, we get even better percentage than 80%. And of course, the large chemical libraries. So Rajit mentioned that ICM has a full support for any libraries. It can be your 50 molecules you're working with. It could be 10,000 molecules of a particular vendor or particular chemistry. So you can read this as DF file or multiple SDF files. You can convert them into all kinds of databases. But most importantly, in the form where you can screen it with multiple means. And you can also take that collection and assign multiple confirmations to those ligands for three-dimensional search through that space. And these are very large spaces, extra large spaces, Wuxi, NMN, Freedom Space. By the way, they're different. And E-molecules was Explore, and purchase of Simple, XTLPi. This is a kind of new company which just purchased LCC previously. We already worked with LCC before. This is a British company. XTLPi is international, but mostly I believe, in China. And we're ready to work with anyone. We will work with you, for example, if you have your own source, we'll convert them into searchable databases. And this very large spaces, they're interesting because they don't, in most cases, they don't overlap. It's enough to have some new chemistry, and you will get into new chemical space with relatively small molecules. I'll skip through that. These are just examples of things we published. This is mostly my academic research, that only representative of Molsoft indirectly, because of course I use all the Molsoft tools at that time. So each of them is an interesting story, but there's no time to go through them. Some of them are repurposing, some of them is multi-target, hitting one molecule with multiple targets. Some of them is finding the first-in-class molecule for some interesting allosteric pocket somewhere far away from the active site. Like this one, for example, we published now the updated version of this ligand, and this is for Alzheimer's, GSK3 beta thing and DIRC1 alpha. So one molecule hits all three, and then we keep improving that, and that's for the tau tau-related Alzheimer's disorder. So these things keep appearing. And in this paper, it's our also we joined forces with Trey Ideker on deep generative chemistry. So I'm not sure it's quite early actually for that. But some of these examples show, like this paper, this antiliminic praziquantel, shows that we can take a new molecule. Like we took praziquantel, and praziquantel is the antiparasitic drug. So then I was approached by these people, and then they say, well, it must do something else in human. And we searched the mole screen, and the mole screen highlighted 5-HT2A, the serotonin receptor. And then, sure enough, when experimentally tested, it was proven that it also hits that receptor. So the mole screen gives you a chance to repurpose an existing drug or existing drug candidate to a new target. And we've done a similar thing with nilotinib versus smoothened receptor in medulloblastoma in the childhood cancer. So now, yeah, this hitting one molecule with one molecule, multiple things. There's a few examples which we published, and this is the actually mutant versus wild type. I'll skip that because you will be, it's too many details. I just want to also highlight at the end that when we use this software in collaboration with experimental startups, with biotech, I'll show you three examples, and in all three, Andy was also involved. These three examples show how we very quickly, from nothing, from visibly impossible challenge. And here the challenge is this. So you have venetoclax, very powerful cancer drug. But unfortunately, there are mutations in BCL2, which it hits, which make venetoclax inactive. So then the task was, can we have something else? But it's such a complicated pocket shape and such a big molecule, it was not easy. But so we identified with ICM the new locations where we can go. We performed the screen against BCL2. Of course, there are also requirements for selectivity versus BCL-XL, and also requirements that we hit the drug-resistant mutants, G101B, and that's the key. And then there's a few other ones over there. And then in 12 months, we arrived at something which has now the name lonitoclax, and it's in clinical trial and passed the phase 1 clinical trial, and it's kind of doing well now in phase 2. So that shows you that in relatively, with just sampling relatively at the end, experimentally testing a small number of possibilities, but going through in that process, going through billions on the back end, we find the solution. And this is all in tight collaboration with experimentalists. That you cannot just do it only on the computer. So that's the patent, and this is where you see the original venetoclax binds here, but we shifted it down below because Molsoft software told us so. We did the pocket finder and we identified the location and we went there. So that's one example. Now another example is the FLT3 for acute myeloid leukemia. There are a number of great drugs. They extremely needed gilteritinib, quizartinib, sorafenib. But the difficulty of FLT3 is that it's a large, relatively large pocket, and then there are mutants which make those ineffective. So there is some mutant which makes FLT3 constitutively active. This is called ITD. And then there is also mutation in this D835 to valine or tyrosine or phenylalanine. And there is another mutation, F391L. So our difficult task was like, find something which will hit those mutants and also will be active against constitutively active FLT3. And we've done that by screening 10 million compounds using the kind of the leak bend. So this advanced docking tools, Enrich and used RGC and N-score, and Andy and Polo were key in that. And this is in collaboration with Lamont Therapeutics. But look at what we found at the end. This is the nanomolar IC50. This is against constitutively active fleet 3 with F691L, 4.7 nanomolar. But if you go to just hyperactive, 0.36 without this mutant, 0.36 nanomolar. This is picomolar ligands. And we gave, that's with the company, we had 163 confirmed hits of 6 chemotypes, which we produce, and 3 series then were further optimized. So, and it spares the wild type. That's another important thing. So the wild type FLT3 is safe. So you can see the ratio of activity. So this is a great candidate. And the last one is this menin balaminib. Okay, before we go, menin. So that now we have now we have 5 now, but I just highlighted 3 of them, which all led to patents and to clinical trials relatively fast. So if you use it correctly, it's a very powerful tool in your hands. But keep in mind that it's not only we need to talk to each other, interact, also we need to talk and interact with the chemist and with the biologist to bring it to success. So this was a long-winded introduction. I apologize for that. I want to now pass the floor to you, and again thank Andy for flying to Bangalore. Of course, it's such a pleasure, as I can say, to Advent, and then all of you. Preet already talked, and Surajit, but also all of you helped, and Aditya solved the little problem we had, just before with the sound. And then, of course, my conclusion cannot go without AI. So I talk to AI, and I can, for legal reasons, I cannot say which of one of the 3 big AIs, basically. Yeah, I asked it to generate the image of a meeting in Bangalore with some, and I gave some names, some of you, of the great users of ICM. And then AI generated this picture. You probably will be curious to see, but I gave a relatively general prompt, give us how it looks like, this great group of ICM users, and here's the image. Now you can look and try to recognize yourself. So AI, of course, may. So is it at least one of you? Is it correct, or all is wrong? Tell me.
All of them are wrong, Dr. Ruben.
All of them are wrong. You see, so that's why you don't rush to use AI, but use ICM. It's not. It's so
What a
expensive. So
lovely way to, you know, end this. Continue, Dr. Ruben, but I am just wanting to tell you that everyone is clapping, when you have spoken about this.
all right. Okay, so wish I were here with you in Bangalore, and let's get started. So, all right.
A heartfelt thank you to you, Dr. Ruben, the founder of Molsoft, for delivering such an insightful and thought-provoking session, and ending with such a humorous and a light-hearted note. Thank you, Dr. Ruben.